In this lesson, we're going to talk about spontaneous generation, Pasteur's experiments, and endosymbiotic theory. So spontaneous generation is the idea that life arises from non-life. And of course, we know that that must have happened at least once in order for life to arise. But spontaneous generation is the idea that it happens regularly and repeatedly. For example, there was a widely held idea that maggots arose from rotting meat when exposed to air, and that you didn't need any flies or anything like that. So spontaneous generation is this idea that life is regularly arising from non-life. There were a series of experiments that were investigating this, and the one that we're going to look at here that finally falsified the idea of spontaneous generation was Pasteur's swan neck flask experiments. So previous experiments had looked at boiling broth and then exposing it to air. So imagine you have a glass flask here and some nutrient broth like chicken broth or something like that in it, and then you boil it and let it cool and it's exposed to air. And what you see is that it gets cloudy because there are microorganisms growing in it. So here we have it's exposed to air. But by being exposed to air here, it's also being exposed to particulate matter in the air. And what was really undecided or unclear before Pasteur's experiments was whether it was the air itself or whether it was something in the air, some sort of particles. And as mentioned before, it was thought that maybe maggots arose from rotting meat just by being exposed to air. So one thing that you could do is you could stop up this flask with a stopper and then boil it and cool it, and that was done. And in this case, there's no growth. However, by stopping this up and evacuating the air out of it, you not only have no particles, but you also have no air. So there's no air and there's no particles, and now it's not clear which of these is making the difference. So Pasteur's swan neck flask experiment, shown here, invokes a clever design of the actual flask in order to sort that out. So again, we've got broth, and the broth is boiled, but now it's in a flask with this swan-type neck, and the neck allows air in, but particles are going to get trapped over here. So now we have air, but no particles, no airborne particles are getting into the broth. And in this case, Pasteur saw no growth. So this seems to demonstrate that it's not just that things generate spontaneously, that life generates spontaneously in broth when it's exposed to air, but you also need this added ingredient of particles that are seeding the broth. And then when you take this a step further and you tip the flask so that you end up with broth encountering that part of the neck where there is particulate matter, and then you tip it back like this, in this case now you would once again see growth, because we can understand now that what's happened is you've encountered the bacteria, etc., that are in here. The particles that have been trapped in the swan neck flask are brought back into contact with the broth. So that's Pasteur's swan neck flask experiment. The clever part of it over here, exposing the boiled broth to air but not to particulate matter, and showing that it is actually the particles that are required for the generation of growth. Let's change gears a bit and look at an important theory in how eukaryotes developed. So we know that eukaryotes have membrane-bound organelles like mitochondria and chloroplasts. And in the 1960s, Lynn Margulis developed a theory called the endosymbiotic theory. And that posits that there was a precursor to a eukaryotic cell. So this is like our pre-eukaryotic cell. And it did not have the abilities that we associate with mitochondria or chloroplasts. And it encountered a small free-living prokaryote. This is our small free-living prokaryote here. And actually engulfed that free-living prokaryote and took it inside itself, and then harnessed that prokaryote's abilities. So, for example, if you want to think of this as a precursor of a mitochondrion, then it would have the ability to perform cellular respiration and liberate more energy from food stores than the pre-eukaryote would, the precursor to the eukaryote. So now by engulfing this and kind of keeping it inside itself, this new hybrid cell with the precursor to the eukaryote and this little prokaryote inside now has the ability to, again, liberate more energy from food stores, so it's getting a benefit. And similarly, this prokaryote that's now living entirely inside the eukaryote is getting fed regularly, kept safe, etc. So that's why it's a symbiosis. It's a symbiosis because both partners are getting benefit, and the endo part is for inside. So once again, a precursor to a eukaryote engulfs a prokaryote that has some abilities like photosynthesis if we're talking about the development of chloroplasts, or cellular respiration if we're talking about the development of mitochondria. And then together they become what we now think of as a eukaryotic cell. That's the endosymbiotic theory about how eukaryotic cells developed. Now let's look at some evidence for that. Eukaryotes have a particular type of ribosome called ADS ribosomes, and that is a measure of how they sediment out when you centrifuge them. It has to do with their density. Whereas prokaryotes have 70S ribosomes, different kinds of ribosomes with different sedimentation rates. And mitochondria and chloroplasts have 70S ribosomes as well, the same as prokaryotes, indicating that this, which might have been a free-living prokaryote, and our idea of what a mitochondrion is, or a chloroplast, have the same kind of ribosomes, and that would make sense if the origin of mitochondria and chloroplasts is as a free-living prokaryote. Similarly, eukaryotes have their DNA associated with proteins, and they keep it in their nucleus. And in contrast, prokaryotes don't have a lot of proteins associated with their DNA, so they have more or less naked DNA, not associated with proteins, and it's in a nucleoid region. Mitochondria and chloroplasts have the same. They also have naked DNA in a nucleoid region, again similar to prokaryotes. Eukaryotes replicate by mitosis and meiosis at the cellular level. Their cells reproduce by mitosis and meiosis, whereas prokaryotes reproduce by binary fission. And mitochondria and chloroplasts replicate independently of their host cells, and they do so by binary fission. So when a cell undergoes mitosis, that's not when it's replicating its mitochondria and chloroplasts. That's happening separately in a process like binary fission. Finally, eukaryotes have a single membrane, and prokaryotes have a single membrane around them. But mitochondria and chloroplasts have double membranes. And that double membrane would have arisen in this process of engulfing, the outer membrane belonging to the former pre-eukaryote and the inner membrane belonging to the former prokaryote. So the double membrane shows the origin in this engulfing process. So that's an introduction to spontaneous generation, Pasteur's experiments, and the endosymbiotic theory.